Thanks for showing up during reading week. I know that's not an easy thing to do. So it's really a pleasure to be here. I want to just have like a disclaimer. I'm not a teaching prof, and I'm not really up on all the pedagogical literature, so I'm hoping that this is kind of like I basically do things by trial and error, and my courses evolve over the years, and I'm going to give you some ideas that I have from my current version of the courses that I teach for how to encourage critical thinking inside the classroom. But I'm really hoping that this causes a lot of, sort of promotes conversation and discussion, and that you can give me some good feedback as well. So with that disclaimer, I just want to say I think teaching is like a really noble profession. It really is like a noble profession. And it's a real pleasure to be able to do it. And it's also, for those of us who enjoy it, it's really fun. And I think it should be fun for the students as well. And it's also a profession where we get to try the same thing repeatedly. So, I mean, in how many walks of life do you get to do the same thing next year, but try to just do it better than you did it this year? And so I think it's really fun to sort of tweak the course and see how you can make improvements. And I try to do that every year by getting feedback from the students. I like to think that my courses sort of evolve by a process. It's an evolution, but instead of mutation and natural selection, it's suggestion and natural selection. So I have ideas that I jot down after every lecture. That worked pretty well, but next year maybe try this. And then I sort of think about my courses during, before this semester comes up, I get into this like high, intense mode of thinking about exactly what I'm going to do. And I also read the student evaluations very carefully, and I have student helpers in my courses who are really crucial. So I'm going to talk to you mainly about my neuroscience course, PNB 2XB3. I have a group of student helpers called NCs, Neuroscience Consultants, basically volunteer undergrad tutors who did really well in the course in previous years. And part of the interview process that I have with those students, which I'm about to undertake next week actually for next fall's class, the students who apply to do that, I ask them, are there ways that you can think of improving the course? And I jot down all their suggestions. And so it's really fun to see how the courses evolve from year to year. But anyway, so what I'm going to talk to you about today is how I try to set the stage in my classes for critical thinking, to sort of let the students know that critical thinking is coming up. So turn your brains on. You're not going to be just able to sit here in the classroom and absorb things passively like a sponge. And then I'm going to talk to you about some of the specific ways that I try to engage them. So one is brain teasers, which are in-class critical thinking exercises. Another is when I take up the homework assignment at the beginning of each class, I have weekly homework assignments, which are also involve a lot of critical thinking. I'm not going to talk so much about the homework assignments today because those aren't inside the classroom. I think we all sort of understand that homework can work really well to sort of keep the students on task and processing the course content. But when I take up the homework at the beginning of each class, I spend really considerable time on that. In my neuroscience class, often a half an hour to 40 minutes, just discussing the homework assignment that they just turned in. And I try to use that as a springboard to sort of dive further into the topic to develop critical thinking. And then if there's time at the end, I'm going to talk about these things called neuromuscular junctures that I do, which are sort of little stretching periods that also involve a little bit of critical thinking. So the way I try to set the stage for students to sort of understand what's going to be expected of them, and I want them to really engage in critical thinking, is in the course syllabus at the very beginning. And this is also an area where it's really fun to be a prof because I think the course syllabus design, the course design is a really creative activity. And it's almost like I think of myself as creating a society. Hopefully it's not a dystopian society, it's more like a utopian society, but it's a society with rules and laws and penalties and rewards. And so it's important to tell the students at the very beginning of the class, what are the rules of this society? What can you be arrested for, like academic integrity violations and things like that? But also, what are you going to be rewarded for? And once I have that sort of framework set up, I try to stick with it very consistently throughout the semester so they kind of understand that this is the reality of that environment that they're in the course. So one thing I mention is at the very beginning, and I go over this in the first lecture, is to do well in this course, you must work diligently and intelligently outside the classroom as well as inside, et cetera, et cetera. And basically, you have to take ownership of your own learning. So you can't just sit there and listen to me lecture and nod and think, oh, that makes sense. You've got to digest that, and you're going to be digesting it both inside and outside the classroom. And I mentioned things like how much you get out of the course depends on how much time, energy, and enthusiasm you put into it. How well and how much you learn is up to you. Okay? I also have the undergrad tutors, the neuroscience consultants, give them advice for how to study for the course on the first day of the course. And then I go over some of the policies that are in the course syllabus, and among them is that cell phone use is prohibited inside the classroom. If they want to use their phones, they're welcome to stand up, leave the room, and use their phone, then come back. And I reference some literature, and if anyone here knows of more literature, I'd be very happy to know of it, but these are research studies that have been done indicating how distracting cell phone use is. It harms the learning process and people, not only the user of the cell phone, but the people around the user. And the same thing for laptop use if it's not for course purposes, if they're on Facebook, if they're checking email. There are research studies that show that that's harmful, so I try to discourage that from the very beginning. And then I also give them a sense of course, I go through all the evaluation components in the course, but one of them that I just want to talk to you briefly about is something called Stump the Prof. So I want them to be asking me questions during lecture, and I want them to not be afraid to ask hard questions. And in fact, I want to encourage probing questions. And when I thought about how to do that over the years, it seemed reasonable to make it actually, to put my money where my mouth is. In other words, to put some of the rewards, and this is 2 extra credit points, for what I really consider valuable in the class. So if a student can ask me a question that I don't know the answer to, which is actually very easy in a neuroscience class. Neuroscience is a huge topic. There are a lot of questions that no one knows the answer to. And there are questions about neuroscience that I won't know the answer to that are known. If someone asks me a great question I think is actually known, so if someone has actually done research on this particular question, a scientist, and there's a known answer, and I think it's a question whose answer I would like to know, then I will nominate that student for a Stump the Prof opportunity. And what that means is that if they go out and research the question, so they find a peer-reviewed scientific article that has the answer to the question they asked in it, and they write a little synopsis of the paper, and they reference it properly, and they send me the paper as well as the published paper as well as their answer, then they can get 2 extra credit Stump the Prof points. And so this serves sort of to let them know very upfront at the very beginning of the class that this is a class where I want their brains to be turned on. I want them to be challenging me if they have questions to ask. I don't want to be afraid of that. It also lets them know that there are things that I don't know. I think that's really important that we as professors let them know that we don't know everything. We're scholars, which means that we're confused much of the time, and we're trying to develop new knowledge. And so we admit when we don't know, and it becomes actually sort of a cause for celebration in the course if someone asks me a question and I nominate them for one of these opportunities. It doesn't happen that often, unfortunately, but when it does happen, the students clap and laugh, and it's like this great thing. And by the way, it also removes a lot of pressure from me. When I first started teaching many years ago, I was afraid that someone would ask me a question on what if I don't know the answer? I'm going to look like an idiot. What am I going to do? This is great. It's like it's completely reverses that to this is a wonderful event. Someone asked me a question I don't know the answer to, and so that's the Stump the Prof, which is just one of the ways of engaging in critical thinking. Because I know that students don't read the syllabus, you can tell them read the syllabus. Read it carefully. They won't read it carefully. So again, I try to put the points on what I actually value. And I realized after a few years, if I really want the students to read the syllabus, even though I've gone through it quickly on the first day of class, why don't I just make the first homework question on the first homework assignment, which is due the second class period, about the syllabus? And so I have a fill-in-the-blank question about the syllabus. And these are things that I just want them to really know in the syllabus. How much you get out of the course depends on how much blank, blank, and blank you put into it. Cell phone use, including texting, is blank in the classroom, which 2 types of recording are not allowed in class, and so on. And so all they have to do is fill in the answers that are in the syllabus for this question. Then I know that they've read it. Another advantage of this is if someone is using a cell phone in the class, I can say, you answered this question on homework 1. You got full credit for that. So you can't claim that you didn't see it in the syllabus. This is about another exercise, which is a term paper called Delve Deeper. I won't talk about that today because it's not in class. Okay. So one of the ways that I encourage critical thinking is these things that I call brain teasers, and I use them in my neuroscience course, which is a second year course, PNB 2X B3. It has about 200 students a year now. I also teach a fourth year course called Bayesian Inference, Psych 4 KK3. That's a much smaller course, about 40 students, and I do a very similar thing, but I call them Bayesian brain bogglers. But these are in-class critical thinking exercises. In the neuroscience course, there's one every class. In my other course, there's at least one every class period. And these pop up at a random time during lecture. So part of the purpose of them is to keep the students on their toes, to introduce an element of surprise in the class so they never quite know what to expect when the brain teaser is going to pop up. I think that helps them just sort of stay alert and engaged in the lecture. The brain teasers are not worth very much. Typically, I make them worth about 5% of the course grade. I find that if it gets above that, the students start getting too nervous about them because I'm asking them to think critically about something that they've just heard in the lecture. So they haven't had time to really study this material yet. This is from the current lecture. When I first introduced these in my first faculty job, which was in Duquesne University in Pittsburgh, I was kind of concerned about that, so I made them extra credit. So I think a good way to introduce new things in your class, if you're not sure about whether they're going to work or not, if it's sort of experimental, is make it extra credit the first year and see how it goes. So after a few years of doing that, actually, I felt really good about them. I made them required. So, yeah. No, not at all. Please interrupt.
I want to do the brain teaser. So how do I tell my student about it? Like, what's the framework shortly that I can use?
Well, what I do is I describe it in the beginning of the class when I go over the syllabus. And then in the very first class period, I give them one, which is not graded. So they actually get to see, basically, they get to see firsthand. They do one for practice, essentially. Okay. And in fact, that's the one I'm going to show you right now. I'm going to show you the actual one. So in the first class period, I go over questions like, what is neuroscience? And I solicit their feedback. It's a study of neurons and nervous systems. And I show them this is a word graphic where the larger the letter, the more frequently the word was used in the title of research articles in the Journal of Neuroscience, which is sort of the flagship journal from the International Society for Neuroscience. And I go over different words, like you can see neuron is the word that's used most commonly. Cortex, mouse is used very commonly, a lot more than human. But then I have, as part of that lecture, why should I care about neuroscience? And one reason is neurological disorders. And so this is just a little video I made based on all the neurological disorders listed in the U.S. National Institute of Neurological Disorders and Stroke from the National Institutes of Health. And I talked to them about this, and it's from A to Z. And this takes 3 minutes. And so I'm giving you an abbreviated version of this because I'm not going to wait all 3 minutes in the interest of time. But the point is I'm just taking them through an introduction to the course, just through the lecture. And then I also mentioned that the nervous system is this fascinating 3-pound universe. It's our universe within. It's everything that's all of our thoughts, feelings, actions, your ability to understand the words I'm speaking right now, converting the sound waves that are vibrating your eardrums into electrical impulses in your nervous system and then processing those to hear words. All that your brain is doing for you. Everything you sense, feel, think, remember, it's all associated with your nervous system. It's due to electrical activity among networks of neurons. So this is sometimes called the 3-pound universe. It weighs about 3 pounds, but it's our internal universe. And I like this graphic. It's a sort of artist rendition of galaxies and stars along with neurons. So it's our universe within. Well, I take them through this, and then I give them a little Q&A. So I'm going to give you that Q&A right now, and then you'll see how the brain teaser sort of pops up. So how many neurons are in the human brain? Does anyone know? All of these students have already taken Dr. Kim's Psych 1XX class, so they actually do know more or less these answers. And so usually someone might say something like 100 trillion, and I say, how many people think that's more or less than 100 trillion? Anyway, it's 100 billion. So it's about 10 to the 11th power, which is about the same as the number of stars in the Milky Way galaxy, in our galaxy. What percentage of the brain do we use? This is a trick question that psych students know. It's 100% of the brain. But there are these myths out there that you only use 90% or 10% or something. You can turn on the other 90% if you just buy my book. So there are a lot of sort of charlatans out there trying to use neuroscience to make money, fake neuroscience. What's the brain's powers in watts? So in joules per second. Just for comparison, a bright light bulb is about 100 watts. For example, a desk lamp might be a 100-watt light bulb. A dim light bulb would be maybe a 60-watt light bulb. 40. Those are good answers, actually. So when I say a dim light bulb is only 60 watts, sometimes the students don't want to go around there, so they'll go to like 100 watts. Well, it turns out that the entire resting power of the body is only about 100 watts. So as you're sitting here right now, you're using about 100 watts. You're burning that much energy. And the brain uses about 20 watts, which is quite impressive in one way, which is that, think of all we do with the brain for only 20 watts, okay? So for only 20 watts of power, we get everything. We get our whole existence, in the internal existence. But it's also impressive in another way, which is that that's 20% of the body power, but the brain only weighs 3 pounds. 3 pounds is much less than 20% of an adult body weight, okay? So proportional to its mass, it's using a lot of energy per unit time. And that's because of all the electrical activity that it's doing. It basically has to keep these neurons charged up like little batteries. And that takes about half of the 20 watts, is just to charge your neurons to keep the voltages different inside the neurons and outside. What's the size of a typical neuron cell body? These are cells like any other cells. How big are they? So are they bigger or smaller than a millimeter? Smaller, because they're microscopic, right? It turns out that a typical neuron cell body is about 10 microns. So 10 microns in diameter, which is 10 millionths of a meter, which is the same as 100th of a millimeter. So 10 to the minus 6 meters is a micron, and these are about 10 microns. Okay? So there are 1,000 microns in a millimeter. So this is about 100th of a millimeter. Anyway, this doesn't come.
When I teach this size thing, I always tell them that what can you see? Because no one actually knows what the smallest object they can see. The smallest object you can see is a neurocell. It's about 100 microns.
What is the smallest object?
Human oocyte is the only cell that's really big in the brain.
Oh, I see. Okay, yeah.
Same size as blood cell.
Yeah. So this comes up a little bit later. I've taken you through a very abbreviated version of the first part of the first lecture of this class. But right after this, I have a brain teaser. And I tell them this is a practice brain teaser, but one of these is going to pop up every lecture. So this is a good one to share with you because it doesn't require any other knowledge of the lectures in the course because it occurs at the beginning of the first lecture. So what I do on the brain teasers is I pose a question to the students. First of all, they have to put all of their notes, computers, textbook away. They just have to have a piece of paper and a pencil and, if called for, a calculator. In this case, there's no calculator required. And then the brain teaser pops up. And this particular brain teaser is how many neurons can fit on your thumbnail, assuming the thumbnail is 1 square centimeter. And the reason I do this is that when you say something like 10 microns, people have no idea what that really means. All they know really is that they can't see it. It's microscopic. So I try to give them a sense of the scale of the nervous system, which is part of critical thinking. Part of it is being able to do order of magnitude calculations, back of the envelope calculations, where you try to sort of put in perspective what's going on. And one way to do that is to try to understand the scale of things, is how many of them would fit in a particular square area, for example. So this is the brain teaser I give in the first class period. And these are fairly structured activities. And so I actually have a timer here, the brain teaser timer. And I show this simultaneously with the brain teaser slide. And usually they have 3 minutes for silent reflection. I'm abbreviating this to 1 minute in the interest of time. And you can see during silent reflection, the prefrontal cortex has a blinking light bulb. So this is sort of partly educational for them just to look at that, although that only, it's only important for them to see that once or twice. Often it's covered over if there's a bigger slide down here. But I show them the timer always. This is a silent reflection period. They're not allowed to discuss anything. And then there's going to be a ping, which you may or may not hear. And at the bell, it's going to change to discussion, to a discussion period. So this is basically a think, pair, share type of exercise. Okay, that would be ping over the loudspeaker. Now it says discussion, and you can see that Broca's area, which is one of the language areas for expressing language in the left frontal cortex, and Wernicke's area, which is a receptive language area for receiving language area, in the temporal lobe, are activated. And the students are encouraged to talk to anyone sitting next to them. They can even jump up and move about this lecture hall if they have someone else in the back of the room they want to talk to, whatever. So it gets completely chaotic in the room, and usually this is another 3 minutes period. So the brain teasers usually are 10 minutes in my class. It's 3, 3, and 4. 3 minutes for reflection, individual reflection, 3 minutes for discussion, where the students collaborate, and then it's 4 minutes for writing in silence, writing their own individual answers that they're going to turn in. So there'll be another ping here. And now this is writing in silence, and the students get really nicely kind of classically conditioned. Once they hear that second ping, it goes from chaos to complete silence like that. It's really impressive. And this light bulb is in the left primary motor cortex, in the region of the motor homunculus, it's called, the representation of the body surface that controls the right hand. So writing, that's the idea. Of course, they're thinking while writing too, so I could have left the prefrontal light bulb on. Anyway, after all this goes, it pings 3 times, and that's the end of the brain teaser, and the TAs walk through the aisles and pick up all the answers, and then those are actually marked. The way we mark them is that they're given either 0, 1, or 2 points. So it's a very simple marking scheme. Basically, 0 means they turned in a blank sheet. They turned in a sheet with just some scribble on it that made no sense at all, or a happy face or a frowny face, or they weren't there at all. Any honest effort, and they get 1 point. So this is largely an attendance mark. I am very much of the mind that I want the students to come to the classroom. I know some faculty say, well, everyone's an adult. If the students don't want to show up to my lecture, fine. It's not fine with me. I recognize it could be fine. It's a different perspective. I put a lot of work into my lectures. I want the students to learn. I'll do everything I can to make sure they're there. Okay? So part of this is I just want them to be there, and I want them to know if they don't come to class, they're going to miss that brain teaser point. And then they get 2 marks if they have the correct answer and they show their work. Okay, so we don't need to let that keep going. I will continue here. So after everyone's turned in the brain teasers, I remind them here that 10 microns is the diameter of a cell body, and 1 micron is 10 to the negative 6 meters. And as a reminder, the question that they were answering is how many neurons could fit into a square centimeter thumbnail, roughly a square centimeter. So this is very simple. This is something that most of them are going to get right even the first day, but it lets them know that I like doing these sort of math problems. So a lot of the questions in my class, not all of them, but tend to be somewhat mathematical. So one simple way to think about this, of course, neurons are more like spheres, but you'll get the same answer if you represent it as a 10 by 10 micron cube. And so since we're talking about just stacking them on a fingernail, we're going to just fill up this 1 centimeter by 1 centimeter, 1 square centimeter, with these 10 by 10 micron, 10 square micron, 10 micron diameter cubes. So I'm losing my, oh, there we go. So 10 microns is 10 times 10 to the minus 6 meters, which is 10 to the minus 5 meters. A centimeter is 10 to the minus 2 meters, which is the same as 1,000 times 10 to the minus 5 meters. So therefore, you can show that there will be 1,000 neuron cell bodies of a micron diameter that can fit into 1 centimeter. So I can go 1,000 across and 1,000 down, which means I'm going to have 1,000 squared, which is 10 cubed squared, which is 10 to the 6, which is 1,000,000. So there are different ways of doing this, but I'm sure if I had given you all 10 minutes and you'd have the chance, 3 of those minutes was collaboration with other people in the room, almost all of you would have come up with 1,000,000 as the answer. So 1,000,000 neurons can fit into your fingernail, just on your fingernail. That, I think, puts in perspective how small these things are. So we have a huge number of tiny things, neurons, in our nervous systems, and this is just the first brain teaser to sort of get that across. Okay, so that's that's an example brain teaser. I have lots of other examples, but okay. I also want to talk about how I use assignments. So at the beginning of each class, this is a 3-hour night class, so they have a week to do the homework assignment, and then they hand it in at the beginning of the next week's class. And I like to give them feedback right away about the assignment, and I like to use the assignment as a way to probe deeper and encourage them to think critically now that they've actually done the assignment. So the assignment itself is really all about critical thinking, but then taking up the assignment, I try to push it further. So just as an example of that, the very first assignment that they do, one of the questions, besides the question about the syllabus, there are usually 5 or 6 questions on the assignment, but question number 2 was that we learned in lecture that the human brain has about 100 billion neurons, and that's an extraordinarily large number. So in order to get a sense for just how large a number that is, answer the following question. How long would it take to generate 100 billion neurons in the brain if the neurons were born at a constant rate of 1 per second? And then the second question is, in fact, the birth rate is not constant, it's exponential because of mitosis. Each neuron, each precursor to a neuron can divide into 2, and its daughter cells each divide into 2. So each generation you would multiply, it goes up by a power of 2, the number of neurons. But to get across just how many neurons there are, 100 billion neurons is a huge number. And just as we have no real concept of 10 microns from our daily life, we don't really have a concept of 100 billion either. So here I'm just converting 100 billion seconds, which is how long it would take to count one neuron at a time for up to 100 billion. I'm converting the seconds to minutes by multiplying by 1 minute and 60 seconds, and then the minutes to hours, the hours to days, and the days to years. And it comes up to 3,171 years. Now, as they work on this question for a week, and there's an online discussion forum, which I call Synapse, because synapses are where the neurons talk to each other so the students can talk. As they work on this for a whole week, almost invariably, some student posts on the Synapse discussion forum, what about leap years? Should I take that into account? And they get all freaked out. Are we going to get marked off if we don't take into account leap years? Well, no, because these are back of the envelope, order of magnitude type of thing. So I'd be happy with like just about 3,000 years. I would accept that for full credit. But I illustrate to the students, because it's almost always come up in discussion on their forum, that taking into account leap years does make some small difference, but not very much. If you take into account leap years, each year is actually 365 and a quarter days, which is why we have a leap year every 4 years to correct for that quarter difference. If you take that into account, it comes up to 3,169. So it's 2 years difference. But that's a fairly big difference, but it's small as a fraction of 3,000. Well, the next question was, since the birth rate is actually exponential because of mitosis, starting with just one precursor cell, if you were to do that, and just keep dividing through mitosis, so each cell divides into 2 daughter cells, how many generations would it take to reach 100 billion cells? Right? Because clearly cells, if they were being added at a constant rate, it would take 3,000 years to get 100 billion neurons. Well, the gestation period, we're born with all 100 billion neurons, by the way. We're born with them. So the gestation period of about 9 months in humans is clearly woefully inadequate we'd have to have a 3,100 year gestation period if we were adding a neuron at a time, 1 second at a time. So it's really because of the exponential growth through mitosis that we're able to get to such a huge number in only 9 months. And in fact, it's remarkable how quickly you can do that. If you just think of one precursor cell, this is of course not exactly what happens, but it captures the essence of what happens. If you have one precursor cell that divides into 2 through mitosis, and then each of those daughter cells, we'll call the initial cell generation 0, each of the daughters is generation 1, then generation 2, and so on. And you can see that the number of cells in each generation is 2 to the power of that generation. So 1, 2, 4, 8. Okay. So we have this sort of exponential growth process.
Go like this, but more like this.
Oh, okay. If you're lucky and good, other people go, but that's exponential also. Just tank. Yeah. Okay. That's an interesting point. Success can build on success. So you could probably model that somehow, and come up with a sort of exponential growth.
Interest costs or returns on like an investment or a loan?
Right. If you have like a 1% return on something, it actually, that doesn't sound like very much, but what is 1% of grows each time, and you get exponential growth that way. That's good. These are different than what I usually get when I ask a question, because the students I have are often, they're in the psychology, neuroscience, behavior department, or in biology, biopsych double majors, and I get a lot of things about bacterial growth in dishes. Population growth is another one. Of course, not all of these things are purely exponential or sustained as exponential. But I like to tell them the story about, there's a myth about how the game chess was begun, which is that there was a wise man who worked for a king way back. And he developed the game chess. He invented it. And the king was so enamored of this game and loved it so much, and all of his court was playing chess all the time, that he wanted to reward the wise man. The wise man was also a bit mischievous, and he also didn't have a lot of money. So he said, or food to feed his family. So he said, oh, I don't want very much. If you could just have your people put a grain of wheat on the first chess square, and then 2 grains on the next, and 4 on the next, and 8 on the next, and so on, until you fill up the board. That's all I want for inventing this game. And the king looked at him sort of strangely. Is that really all you want? I mean, don't you want more? No, that's all. So he said, okay, and he sent out his people or his person to just get a little wheat and fill up the chessboard. And the answer came back that actually that was going to be difficult. There wasn't enough wheat in the kingdom to fill up the chessboard, even though the king had lots of fields of wheat. And that's because this is an exponential growth process. So if you call square, the square number here, 1 through 64, there are 8 by 8 squares here, so 64 total. And then the number of grains that you need to put on that square is 2 to the n minus 1. So for example, the first square has 2 to the 0. The second square has 2 to the 1, so it's 2 to the 2 minus 1. Okay. And I'm just going to go through here. We can see, we know that at 37, we're going to be past 100 billion grains of wheat. But we have to go to 64. So this is really something. It's just one chess square, but here we go, we're approaching 37. This E means the power of 10, so we just passed 10 to the 11 here. Now you see we're at 10 to the 13, 10 to the 14 by square 50. These are huge numbers. These numbers are so big that I didn't know what they were called. I didn't know what came after, like whatever. I mean, even 10 to the 15 is very high. So it turns out to be about 10 to the 19, almost 10 times 10 to the 18. This turns out to be the same. So this is 2 to the 63rd power. That's just a number that needs to go on the last square. You also have to add all the other numbers. This is just a number on the very last square, the 64th square, so 2 to the 63. Turns out that this is 10 quintillion. 10 to the 18 turns out to be called quintillion. So you need 10 quintillion grains of wheat. And if you do a back of the envelope sort of order of magnitude calculation here, you can show that if you assume that, I looked this up, I think reasonably speaking, 10 square millimeters in area is about right for a grain of wheat. So 10 quintillion would occupy 10 times 10 to the 19 square millimeters, about 10 to the 20 square millimeters of wheat. It turns out that that's enough to cover the entire surface area of the Earth, not just the landmass, everything, the oceans, with wheat, with no space in between. Okay, so this one chess set, this is enough to literally cover the globe with wheat. So it's not a surprise that there wasn't enough in the kingdom. So you can show that because the Earth's radius is about 6,000 kilometers. So its surface area is 4 pi r squared, which is about 10 to the 20 square millimeters. And that's what we have here, 10 to the 20 square millimeters. Okay. So this may seem far from neuroscience, but I'm trying to develop a sense in the students of how to reason with numbers. There are a lot of numbers, there's a lot of physics, chemistry, and neuroscience, and I want to sort of encourage this type of thinking. So we have a lot of questions like this. Not all of them are like this in the course. There are a lot of brain teasers on other things and a lot of homework on other things. But then I tell them that my favorite example is croissants of exponential growth. And I tell them that they've learned about brain sections already in the previous lecture. There are different sections. You can slice the brain in different anatomical planes, and this is called a coronal section. And so I tell them at this point that I don't want to freak them out. I don't usually show real sections of things, but I'm about to show them a coronal section of a croissant. Okay, and I'm going to show them. But anyway, so here you have the way croissants are made is that if you look in a typical cookbook for pastries, it's basically to make a business envelope, a trifold, and then roll, and then do a trifold again and roll. And you do this typically about 6 times. So you end up getting, every time you do the trifold, you get 3. So you end up getting 3 to the 6 or 729 layers. And that's exponential growth. This is where I give them the coronal section. This is a section through the croissant, and you can see all these layers. You can't see all 369 of them. But this is why they're so flaky and delicious, croissants. Anyway, that's just an example of that's the very first homework assignment and how I take it up in class. And as they get further into the course, these get a little more technical, but it's the same idea. I try to sort of push them to think critically during the homework review. And then I want to talk now about, and this is good, I have time. One final thing I do during class, because it is a 3-hour night class, and I have a 1 10-minute break period. But then because the brain teaser happens, it's sort of also a break from the lecture. And I also have a sort of get up and stretch break. And I know that this is, people have talked about this before. I think Joe talked about this during Joe Kim, his MacDUET presentation, the value of exercise. And there are research studies on that now. I don't know how valuable this is as an actual exercise, but my intuition is that it seems to help. And it's also kind of an educational opportunity, kind of a little critical thinking. So what I do is at a random time period for them, they don't know when this is going to happen, I say, okay, let's take a neuromuscular juncture. First of all, this is a pun, because where neurons connect to muscles, where they release acetylcholine neurotransmitter to muscles, is called a neuromuscular junction. That's the synapse between a motor neuron and muscle. So this is a neuromuscular juncture. And neuroscientists, by the way, are kind of famous for puns. So I also pepper my whole talks with puns. And I can't take credit for this pun. This pun was actually suggested to me when I think I may have mentioned neuromuscular junction to Dita Gillespie in my department, who also happens to be my spouse and is also a neuroscientist. And she's an inveterate punster. She's the one who converted me to a punster. And she said, why don't you call it a neuromuscular juncture, because it's a point in time. So we take a neuromuscular juncture, which starts as a free stretch, but then I give them a challenge to incorporate into their stretch. And so it usually looks something like this. Use these neurons to activate these neurons. And this is appropriate to the class that the lecture of the week that we're doing, the topic. So they already know that these neurons are in the motor representation in the left hemisphere, and this is the hand area. And it's the left hemisphere, and things are crossed in the nervous system. The left hemisphere controls the right side of the body. So they know they have to move their right hand. That's what this is. But they need to activate these neurons. How do you use these neurons to activate these neurons? These neurons move your right hand. These are the visual cortical neurons in the occipital lobe. And they've already learned that the visual cortex on each side of the brain pays attention to the other side of the world. So the way to activate these neurons with these neurons is to move your right hand on the right side of your body, something like this. And then you'll activate those neurons. And so they tried that for a while. Just to illustrate, here though, this is the motor cortex, and this is the primary visual cortex in the left hemisphere, which are associated with the right side. This is the motor homunculus, we call it, the representation of the body in the motor cortex, and this is the hand area. They've learned all this before. So they need to put it together, and they need to basically wave with their right arm. So there's one of these pops up now in every lecture as well, and it's just a brief period, and then we sit down and resume the lecture, but their blood has gotten pumping, and I sort of think of it as I try to keep them on their toes during the lecture, the brain teasers, all that, and now they're like literally on their toes during the stretching as well, which I think is also useful. And that, I think, is all I had. So I'm happy to open the floor to questions and so on. Thanks.